Introduction. Welcome to today's lecture on the topic of amino acids and their role in metabolism. In this comprehensive discussion, we will focus specifically on arginine, one of the 20 essential amino acids. We will explore its structure, properties, and essentiality, as well as its involvement in various metabolic pathways. Furthermore, we will delve into the biosynthesis and catabolism of arginine, its transport mechanisms, and its crucial role in the synthesis of nitric oxide. Additionally, we will investigate the relationship between arginine, nitric oxide, and cardiovascular health, immune function, cancer, wound healing, and its potential implications in neurological disorders. By the end of this lecture, you will have a thorough understanding of the diverse aspects of arginine metabolism and its importance in human physiology. Amino acids are the building blocks of proteins and play an indispensable role in various metabolic processes within living organisms. They are crucial for protein synthesis, cellular growth, and tissue repair. Additionally, amino acids act as precursors for the synthesis of various biomolecules, including hormones, neurotransmitters, and nucleotides. Furthermore, they contribute to energy production and participate in the regulation of cellular signaling pathways. Out of the 20 amino acids, arginine stands out due to its unique structure and versatile functions within the human body. Arginine is a semi-essential amino acid that contains a guanidine group in its side chain. It is a basic amino acid with a positively charged guanidinium ion, which imparts distinct chemical properties. Arginine is vital for proper growth and development, immune function, and wound healing. While the majority of amino acids can be synthesized endogenously, arginine becomes essential under certain physiological conditions, such as during periods of growth, stress, or in individuals with specific genetic disorders. Arginine is metabolized through various pathways in the body. These pathways include the urea cycle, the arginine-proline pathway, the arginine-creatine pathway, and the arginine-nitric oxide pathway. Each pathway has its own unique set of enzymes and reactions that regulate the production and utilization of arginine and its metabolites. Arginine is synthesized from citrulline via a series of enzymatic reactions in the urea cycle. This cycle takes place mainly in the liver and plays a critical role in the detoxification of ammonia, which is generated during amino acid metabolism. The urea cycle involves 5 key enzymes, including carbamyl phosphate synthetase, ornithine transcarbamylase, argininosuccinate synthetase, argininosuccinate lyase, and arginase. Together, these enzymes convert citrulline into arginine, ensuring the continuous supply of this essential amino acid. When arginine is not utilized for protein synthesis or other metabolic processes, it undergoes degradation through the arginase enzyme. Arginase cleaves arginine into ornithine and urea, therefore linking arginine metabolism with the urea cycle. Ornithine can then re-enter the urea cycle or be further metabolized in other biochemical pathways. The cellular uptake of arginine is facilitated by specific transport proteins located on the plasma membrane. These transporters allow the efficient delivery of arginine into cells, enabling its utilization in various metabolic processes. Additionally, the concentrations of arginine transporters can be regulated under different physiological conditions, thus influencing the intracellular levels of arginine. One of the key functions of arginine is its conversion into nitric oxide, NO, through the action of nitric oxide synthase, NOS, enzymes. NO is a potent signaling molecule involved in numerous physiological and pathological processes. The production of NO from arginine plays a significant role in vasodilation, regulation of blood pressure, modulation of immune responses, and neurotransmission. The interplay between arginine, nitric oxide, and cardiovascular health is of utmost importance. Proper endothelial function relies on an adequate supply of arginine, which serves as a substrate for NO synthesis. Nitric oxide, in turn, plays a pivotal role in vascular homeostasis, maintaining blood vessel integrity, and regulating blood flow. Dysfunction in the arginine-NO pathway has been implicated in various cardiovascular disorders, including hypertension, atherosclerosis, and heart failure. Arginine plays a critical role in modulating immune responses. It is involved in the proliferation and activation of immune cells, such as T lymphocytes, macrophages, and natural killer cells. Arginine metabolism influences the production of cytokines and the generation of reactive nitrogen species, both of which are essential for immune cell function. Furthermore, arginine availability can impact immune cell polarization and the effectiveness of immune responses against pathogens. The relationship between arginine and cancer is complex. In some cancer types, arginine deprivation has been shown to inhibit tumor growth by depriving cancer cells of an essential nutrient. On the other hand, certain tumors exhibit enhanced arginine uptake and metabolism, enabling them to utilize arginine as a source of energy and promoting their survival and proliferation. The intricate interplay between arginine availability, cancer metabolism, and the tumor microenvironment requires further exploration for the development of targeted therapies. Arginine plays a crucial role in various stages of wound healing. It is essential for the synthesis of collagen, a key structural component of the extracellular matrix. Arginine promotes angiogenesis, the formation of new blood vessels, which is crucial for adequate nutrient and oxygen supply to healing tissues. Additionally, arginine modulates immune responses during wound healing, facilitating tissue remodeling and regeneration. Emerging evidence suggests that arginine and its metabolites may have implications in neurological disorders. Nitric oxide, derived from arginine, is involved in neuronal communication and synaptic plasticity. Dysregulation of arginine metabolism may contribute to neuroinflammation, oxidative stress, and impaired neurotransmission, all of which are implicated in neurodegenerative diseases such as Alzheimer's and Parkinson's disease. Further research is necessary to unravel the precise mechanisms underlying the involvement of arginine in neurological disorders. In conclusion, arginine, an essential amino acid, plays a multifaceted role in human metabolism. Its involvement spans various pathways, such as the urea cycle, arginine nitric oxide pathway, arginine proline pathway, and arginine creatine pathway. Arginine is crucial for cardiovascular health, immune function, wound healing, cancer metabolism, and potential implications in neurological disorders. Understanding the complexities of arginine metabolism provides a foundation for further research aimed at harnessing its therapeutic potential and developing targeted interventions.